Hello, my name is Simon Dawson, and I'm a junior working in the Queller-Strassman lab. For the past 2 semesters, I've been working with the bacteria strain Parabolidaria banya. This bacteria is commonly found with the social amoeba Dictyostelium discoideum, and within this relationship, banya is inedible and able to infect D. discoideum. Not much is known about P. banya's infection mechanism, but based on the microscopy assay shown in the bottom of the introduction section, where banya seems to aggregate around the D. discoideum cell, it's possible that the flagellum is a necessary part of its infection mechanism. That is reinforced by the fact that the close relative Burkholderia haleala uses chemotaxis to find D. discoideum host cells. So all this preliminary information led me to my hypothesis that the removal of P. banya's motility should reduce its ability to infect amoebae. But to test this, though, I first need to create non-motile mutants. To accomplish this, I had to choose a target within the bacteria's genome, and I chose the gene flagellum C. This gene is responsible for the filament that is used in creating the flagellum, and removing it should make the mutant non-motile. To create this mutant, I first used Golden Gate assembly to create a custom plasmid that consisted of a PCS1 backbone and a kanamycin resistance gene in between two stretches of DNA called homologous regions that corresponded to the two sequences of DNA on both sides of the flagellum C gene in Parabolidaria banya's genome. Then I used an E. coli donor to conjugate the plasmid into a strain of Parabolidaria banya. At this point, homologous recombination occurs, and that flagellum C gene is swapped out with my kanamycin resistance gene. The PCS1 backbone is a suicide vector, so it should cease to function on its own accord after the homologous recombination. But to make sure that it does, I used an Hyco tetracycline counter-selection plate to check. Then the insertion of the kanamycin resistance gene was checked using custom PCR primers, and I was able to then finally check the mutant's phenotype with a 0.3% LB motility assay. I also conducted the motility assay with a wild-type strain of banya and a known non-motile transposon mutant, flagellum S. The photos at the bottom show the motility assay after 24 and 48 hours. While the flagellum S mutant, as expected, shows no movement, the flagellum C mutant shows a reduced but not completely eliminated motile phenotype. It moves slower than the wild type, but surely more than the flagellum S mutants, which isn't exactly what I wanted with my final phenotype. Since I know that the gene was properly replaced, it's clear that this just wasn't a good target, as there must be some sort of alternative source for making the flagellum. But this data is still really important to my overall hypothesis because this shows that my procedure is a reliable way of genetically modifying Parabolidaria banya. My next step is to redo this procedure, except with a new target. I want to target a collection of 3 genes, flagellum L, flagellum M, and flagellum N. These genes are all structural and should be reliable targets for removing the phenotype. Additionally, once I have that non-motile mutant, I plan on testing its new relationship with Dictyostelium discoideum using a gentamicin protection assay and a microscopy assay. But that is where I am so far in my research. There's still a lot for me to do, and I'm excited to continue working on it. Thank you for listening to my talk. If you have any questions or would like to talk to me about the project at all, I'll be holding a Q&A session over Zoom at 3 p.m. on Thursday, April 21st. Thank you again.